today we will present you our laboratory of adaptive brain stimulation. This is one of two non-invasive stimulation laboratories in the Center for Cognition and Decision Making. Here you can see our equipment for transcranial magnetic stimulation Nexstim, which allows to non-invasively stimulate brain cortex. It includes infrared camera, the screen which allows to run a number of behavioral tasks, a memory-navigated system, an amplifier for image recording, and stimulation coil with figure-of-8 configuration. TMS coil allows it to either activate or deactivate different brain regions depending on the stimulation parameters. TMS stimulation is accompanied by a specific clicking sound, which makes it necessary to use earplugs or noise-suppressing headphones. Now you can see a marker with light reflecting coating, which is attached to the head of participant. This special marker allows infrared camera to track the special location of participant's head. To conduct an MRI-navigated stimulation on Nexstim or any other TMS devices, they have to correlate an individual MRI scan with the real participant's head size and his real parameters. Using a special Y-stick, they correlate points on the participant's head with the same points that they previously located on an individual MRI scan, which means that future stimulation will be local enough. To record electromyography, we will use bipolar montage, which means that the electrodes are attached to a little finger and a thumb. Here you can see abductor pollicis brevis muscle, which abducts the thumb, and here there is a location of the abductor digiti minimi muscle, which abducts the pinky. If they ask the participant to perform a movement, they can observe a corresponding activity on the screen. We can observe similar activity from the leg muscle if they ask the subject to strain his leg. Let's put on the headphones and start our experiment. Using the Nexstim, we can perform brain mapping. For instance, they can do the mapping of the motor cortex. If they stimulate motor cortex in the left hemisphere, we will observe the response from the contralateral hand or leg and vice versa. Now we can try to stimulate the cortex. We will not consider in detail the entire mapping procedure. We just want to stress the point that we can do the procedures with non-invasive brain stimulation, which we previously done only invasively by neurosurgeons. Similarly, what they are doing now by stimulating different cortical areas, they are demonstrating the somatotopic gradient, drew the homunculus gradient for healthy participants.
Somatotopic или градиент вот этого гомункулуса нарисовать у здоровых.
Brain mapping is usually carried out in several stages. Firstly, the rough mapping is performed to find a muscle hotspot, where muscle response is the most prominent. Then, the motor threshold is defined, which is individual for every participant. The maximum power of stimulation could be up to 2 Tesla. Therefore, usually stimulation is conducted using 30–40% from the maximum power. TMS is used for research and diagnostic purposes as well as treatment. It causes minimal discomfort. Now you can see the hand twitching when the stimulation is performed using higher intensity. Here you can see location of the spots with the most prominent responses to stimulation, green for thumb and brown for pinky. According to the homunculus, we observe more responses from the thumb laterally. Let's see if they have responses from the leg muscle. Here we can see the response activity. That is pre-stimulant activity and motor work potential. The equipment of Adaptive Stimulation Laboratory for transcranial magnetic stimulation allows to investigate cortical states and connection within the brain. Additionally, they have special equipment to conduct biofeedback training in the laboratory. Myographic signal is sent to the special software, with each visualization you can see on the screen. Using the software, participant understands how successful he or she is in motor task execution. Participants were instructed to contract the thumb muscle and simultaneously to relax the pinky muscle.